The future of Septerna. Alrighty, so our next presenter, we're going to be doing online, and then we have somebody live to do the question and answers. A couple weeks ago, or probably about a month ago, these two came to us and said, you know, we have this new gene therapy thing that we're working on, and we wanted to see if you'd be interested in hearing about it. And we listened, and we're both, Michelle and I are like, oh, you know, let's see what's going to happen with this. So we're very excited to show you a recording from Bijou from Rutgers, who's going to talk about it, and then we're going to have Dr. Nikki Partridge, come up and do some Q&A with you. So.
Hi, everyone. My name is Bijou Parkhan. I'm a professor of biomedical engineering. Wish I could be with everyone at the conference today, but I was here to share a little bit about what might be on the horizon for patients with hyperparathyroidism, which is the use of a new experimental treatment that's referred to as gene therapy. So I thought we'd dive right in, and by starting with what is a gene and what is gene therapy? Well, genes are the DNA sequence that kind of encode for a protein. And sometimes those sequences don't have the right kind of letters in them. And so the protein that comes out doesn't have the same function that it's supposed to, and can oftentimes cause some sort of dysfunction in tissue and kind of health. So the concept of gene therapy is to try to introduce the original version, the non-mutated version of that DNA sequence, to make the corrected protein and hopefully restore a person's own function and phenotype. And so what we're going to dive into in the next number of slides is what are some, kind of examples, what's the history of this field, and how does it pertain to hypoparathyroidism. So we'll start with just kind of a little bit of background about this, um, you know, history of gene therapy and see where it's being used currently and what we like to see it used in the future. So gene therapy, as we kind of originally discussed, was designed for cases where there's kind of a mutated DNA sequence that we'd want to correct. And so there are a number of monogenic conditions where a single gene is leading to a single protein that's not in the right configuration. And this is a great sweet spot for gene therapy. So some examples are shown here, indications of the eye, of the central nervous system, of muscle, where one mutation in one protein is causing a pathophysiology in these specific conditions. And there's been a lot of exploration of gene therapies to correct these kind of single mutated sequences by delivering the gene with the correct sequence back to this person. And how these genes are being delivered is there are many different forms that people are evaluating, but certainly the most widely used one, where there are actually clinical products that are gene therapies that are, using this format, are referred to as AAVs. These are adeno-associated viruses. They're destabilized, so these are viruses that do enter into cells, which makes them very useful for delivering DNA to cells as a gene therapy. But they don't come with all the um, issues of growing like as viruses do or causing any sort of pathogenesis. They've been destabilized and really exploited for their ability to enter into human cells as a tool to get genes and genetic structures that we can make in the lab packaged into these viruses and ultimately, delivered as a treatment. What's nice about AAVs is that they've been around for several decades and have been explored in terms of how long they can express a gene in the body. And typically, they can be 1 to 3 years. There's certainly exceptions to that rule. They have relatively low immunogenicity, but that said, they are still viruses. The body still does recognize them as foreign, and that's something that needs to be managed whenever evaluating a gene therapy. Another thing that's very useful for AAVs is they come in different flavors, different serotypes, And these different serotypes, for example, AAV2 has a better tissue tropism, i.e., it can be delivered to a particular tissue with much higher affinity than other tissues. So you can kind of pick and choose your AAV serotype for the tissue of interest that you want to deliver your gene, in, whether it be muscle or the central nervous system. Different serotypes have different uh, favorable uh, delivery to those tissues. So AAVs is kind of what package and deliver genes into cells. And what we're showing here is over the last decade or so, a number of now FDA approved products using AAVs or certain types of viral vectors in order to deliver a gene to the human body. And some of these cases are in eye disease, some are even in cancer, where they're genetically modifying immune cells. But all told, there are now several gene therapies that are available on the market. One thing that the field is learning is it's certainly expensive to make, these kind of new forms of medicine. But in many of these respects, these are curing these uh, different indications with a single treatment. And that can be the potential and the power of gene therapy. With one single treatment, we could correct a mutated protein, in theory, for a long period of time, and kind of reduce healthcare burden in that way. These are some of the early uh, uh, breakthroughs that occurred a decade ago. If we fast forward now in terms of how many treatments are available that are considered gene therapy, we have over 100 if we include the delivery of DNA or RNA or even genetically modified cells. And there are thousands more that are being evaluated. So it's a real emerging field. A lot of examples of successes, and that's been exciting. But there's more to go, more to learn, uh, cost to be reduced, and also more evaluation of it for new indications. This is kind of where our work comes in, and others, where we're starting to evaluate gene therapy, not just for the expression of a protein inside of a cell, which all of those kind of previous conditions that I was describing in the eye or the muscle or central nervous system, those were conditions where a mutated DNA sequence led to a mutated protein, but that protein existed inside of the cell. And so conventional gene therapy would try to, correct that protein inside of that individual cell. What we and others are evaluating is, well, could we make a protein that is missing in someone, but that's a secreted protein? So instead of this cell expressing this gene and keeping that gene product, the protein itself, inside of itself, now that cell would be, expressing that gene to make that protein, but that protein is destined outside of it itself. It's being secreted, and it ultimately will kind of enter into the bloodstream and reach levels that the entire body might experience. And so that's kind of where we're evolving our work, and how it ultimately will relate to its use or potential use in an area like hyperparathyroidism. So let me transition and kind of talk a little bit about what gene therapy does in this format, where we're looking at a secreted protein, entering to the bloodstream. What does it look like? And ultimately, what is the pharmacology of this medicine, which is very different than, an aspirin or, something you might take for, an asthma inhaler. This is, a very different type of medicine. It acts and behaves differently. And so one area of interest is how to predict um, what the levels are of a gene therapy by, administered by an AAV. So what does that kind of look like from administration to expression? Well, we start again with DNA sequence for something that, a protein that we want the human body to express. For example, something that's been missing. And so that gene, that therapeutic protein, is being driven by what's called a strong promoter. All right. So this is effectively a signal in DNA to tell the cell to always make this protein at some stable kind of rate. So that piece of DNA is through some molecular biology connected and inserted into the DNA of an AAV of a virus and packaged in a way so that the virus now is sort of delivering this gene to cells along with the other genes it needs to create kind of that viral shell. So these AAVs, these viral vectors that are carrying now this gene therapy specifically, um, in many respects can be administered locally, but what we're envisioning is an intravenous administration, a one-time infusion, where in large part the viruses enter into through this administration route, they enter into the liver, and the liver then uptakes these viral vectors and starts to actually produce or express that gene therapy. And if it's a secreted protein, like where we will discuss for the rest of this talk, that protein will start to be secreted by the liver itself and ultimately accumulate and found in hopefully appreciable levels in the bloodstream. And so, as you can see, from administration to gene expression, there are a number of important steps. But ultimately, what's you know im- very critical to have a bit of control and precision on is the level of this gene therapy being expressed in the protein that ultimately gets secreted into the bloodstream. So pharmacology is really a way using mathematics to have a better predictive sense of what those blood levels of that gene therapy would be in order to kind of uh, produce it in a range that is both safe and effective for a given therapeutic protein. So that's kind of where we want this work to head to, something that has a good amount of prediction of if we give a certain amount of gene therapy, this is how much protein we would expect in the bloodstream. I'll share with you kind of a little bit of an example of this type of data and the type of pharmacological modeling that we have done in the lab. So here's an experiment where we're looking at mice. They were administered two different doses of a gene therapy that's expressing a secreted protein. So the red line has a higher dose than the black line, and this protein that's being released again, it's entering into the bloodstream, and we're taking weekly blood samples to measure the amount of this protein. And what you can see is that after a number of weeks, if we look to about 28 days, kind of right in the middle of the graph, about 4 weeks, the protein that's being secreted, it reaches what we refer to as a steady state blood concentration level. You see it kind of gets flat and it sort of stays flat for the remainder of the time that we're making these blood measurements. And it turns out that steady state concentration, if we compare against the different doses of gene therapy that we administer to the mice, it turns out that there's a very good dose response, meaning that the more of these gene therapeutic viral vectors that we give to the mice, the relatively higher there is of this steady state blood concentration. Perhaps as to be expected, but we're finally kind of seeing some experimental evidence that there's some relationship between these two, and is often a sign that there could be some kind of predictable mathematics that describes that relationship. Um, Another thing that we learned and also confirmed in this study, as many others have, is by administering these gene therapies in mice and studying the mice after several weeks later, we could confirm that the liver, in fact, was by order of magnitudes more the tissue where the gene therapy was being expressed. So we looked at all the different tissues, measured, the gene that we were administering to the mice, and when we found it in much higher percentages in the liver. So this should kind of round out some experimental data that kind of provides you a little bit more confidence that the liver is a primary site of intravenous kind of gene therapy, and that the steady state blood concentrations of a secreted protein from a gene therapy, there's some dose dependence, and what we did in to kind of have a more mathematical view of this, and I won't dive so much into the details, is we looked at this phenomena and used really mass conservation laws without going into great depth. What we eventually kind of landed on is that at steady state, when the blood concentration is at a certain level, that must mean that the production coming from the liver must equal what is being eliminated for this protein. So usually proteins are eliminated by, either through the kidney, through the liver, through the spleen. Um, some shape or form, they're um, being reduced from the blood, and at steady state, there's kind of a balance of the two. So the liver's producing it, and the protein is being eliminated, and it's that balance that keeps the level flat. And so using this kind of set of mathematical equations, um, along with experimental data where we would measure the secretion rate of a cell, we can kind of stitch these two things together, a cell secretion rate, along with this mathematical model that can take that secretion rate and scale it to a larger subject like a mouse. And we actually showed with a known protein that we'd worked with many times, this luciferase, in comparison to an unknown protein, PTH, that we'll certainly dive more into in a moment, that we can measure that secretion rate, and ultimately with PTH, actually show that we can simulate the blood concentration that we think it should be based on this math model and some experimental data like a cell secretion rate. And actually, with that level of simulation, we can choose doses. What's shown in red are the two actual doses that we chose to study in mice. We administered them in mice, and we measured the level of PTH in these mice at steady state. And it turns out that our prediction and our actual data were extremely close, with about a 5 picogram per milliliter error between the two. So it was a good sign that there is a mathematical relationship here between gene therapy dose and blood concentration levels, and that we can use this math model to actually predict unknown new protein therapies that we might want to deliver. And so the hope is that this will be able to accelerate research in the field, minimize the use of animals, use of additional subjects, um, and help researchers get into a range of a protein therapy in the blood that they're supposed to be in to keep things safe and effective. That's kind of the power of pharmacology. All right. So, now I want to kind of just dive into why we're all here, which is the actual encoding of parathyroid hormone within a gene therapy. So my lab, along with Dr. Parker's lab, who is here with you in the audience to help field some questions, we took on this challenge of trying to create a gene therapeutic version of parathyroid hormone. This is a very critical hormone released by the parathyroid glands, and PTH has wide actions on several tissues in the body, kidneys, bones, um, and in patients with hypoparathyroidism, uh, there could be many variations of the theme, but either they are not producing enough PTH, or that the body's kind of insensitive to the action of PTH, creating this physiology imbalance that PTH is very important for minerals like calcium and phosphate, and without that type of signaling, there's kind of health concerns that emerge from that. So several groups have made recombinant PTH protein as kind of a supplement to take. There are some that are already available and marketed, and certainly a number of others in development to supplement patients with PTH. Our question was, can we make PTH in a gene therapy where you could administer it in theory one time, and your own body would then produce PTH again, and hopefully with some level of control and with the right type of bioactivity. And so Dr. Partridge and I kind of entered into this journey, and we started by looking at PTH at the genetic level, and realizing that it has a number of different precursor kind of proteins. The first of which that cells make is this pre-pro PTH. And eventually, cells will cleave this down to the shorter PTH hormone itself that is found in the body. Um, we started with that gene structure just to be comprehensive and expressed that gene in hepatocytes in cell culture. And we showed that liver cells can actually make PTH in this gene therapy form, where normally they don't make a protein like that. So as a sign that we could get PTH to be expressed by non uh, parathyroid gland cells, and gave us a little foothold to build from. So from there, we took this AAV PTH gene therapy and we applied it to mice, intravenously administered it to mice. And at higher doses, we could see first and foremost that, again, these viruses were entering into the liver, as shown here in a dose dependent way. But more importantly, at high levels, we could administer these viral particles and see now in the bloodstream of these mice weeks later that PTH was detectable at pretty high levels. And not only was it detectable at high levels, but in the high dose groups, we looked at those mice, and these are wild type mice. There's you know nothing particularly different about them, um, but they the ones that are getting this high dose gene therapy, they're certainly making more PTH than their counterparts. So that high level of PTH in the animal's bloodstream, when we start to look at the bones of these animals, if you recall, PTH kind of acts on bones. We saw that there was, more substantial bone growth, and this again was a sign that not only were we making PTH, but that it was actually having the activity that we thought it should. In this example, kind of increasing some bone mass. So this was kind of our initial, you know, uh, research uh, publication describing this work. There's certainly a lot more to it, but we feel like it was kind of a proof of concept that we can make PTH as a gene therapy. And now I just want to kind of end with what we again hope to see kind of in the future with having some better control of it. Now, PTH itself actually fluctuates over the course of a day. So we like to be in this kind of tight range of expression of PTH, but in reality, we might want to even better mimic this dynamic of fluctuation to again match a person's own physiology. And so, how can we do that from a gene therapeutic standpoint? Well, it goes back to something that I mentioned before, which was playing around with this promoter. So the promoter right now is a constitutive promoter that we use. It's always on, turns the light on, it stays on. But there are clever ways to actually make those promoters turn on and off, almost like a switch, like a light switch. And this is where we think, in addition to PTH, controlling the promoter to make it even more physiologic may have improved benefits. And so I'll give you some examples, and we'll kind of end here. This is an example of instead of a constitutive promoter, here we made a circadian promoter. So this promoter takes a gene therapy and turns it on and off over the course of a day based on the light cycle, based on the mouse's own circadian rhythm. This was a paper published several years ago where we showed the circadian gene therapy in cell culture and also in a mouse model. And we and others have done a lot more in this area. What we're trying to aim for in terms of real kind of visionary work is can we make this type of gene therapy control with wireless control? And that's where things are headed. Where in this case we have kind of a mechanical gene switch that, in theory, we can turn on by push of a button to kind of release a gene therapeutic protein, for example, PTH, maybe after a meal in a push button way using an app and kind of a wearable device. So this is where we hope the field is headed. With that, I'm just going to summarize and say that this is an experimental treatment is not available for humans yet. Hopefully on the horizon with a lot of work and certainly a lot more evaluation um, to be done. We think this aspect of pharmacology of modeling gene therapy is going to be very critical for secreted proteins like PTH and others. I think we've shown that PTH can be delivered and be bioactive in mouse models, and we're hopefully going to scale this up over time. And we think that there are gene switches out there, and choosing the right one to control PTH better in the body may lead to an overall, uh, uh, more physiological impact and something that lasts a lot longer. So with that, I'm going to end and say thank you and have a wonderful conference. Again, wish I could be there, but you're in great hands with Dr. Nikki Partridge to answer questions and represent Rutgers. Go Texas.